But we're going to move on to our second company of the evening. So I have tonight with us Dr. Stephen Pierce, and he is the CEO of a company called EMTensor.
Okay, and thanks again to you, Michael, for the help that you've provided. This is a vastly improved presentation compared to what it was before. So I'm the CEO of EMTensor, and we're in the business of accelerating brain injury treatment. We hope to save millions from death or disability and avoid much of the $1 trillion bill per year in global ongoing disabled care costs. Globally, affecting 17 million people per year, stroke is the leading cause of disability and the second leading cause of death. You heard about the first leading cause during the last presentation. Early stroke determination dramatically improves outcomes using existing medical therapies. Yep, everyone that's needed is already in existence. Typical times to treatment in the best case of cities remain about 4 hours because the patient must travel to obtain a confirmatory image in a hospital CT or MRI. Reducing that time to treatment to under the golden hour could eliminate disability from 100 million stroke victims in the coming decade and save the lives of more than 2 million others per year. Accelerating time to treatment for all stroke patients from 4 or more hours to minutes can turn back time on disability and death. Our portable brain imager moves the confirmatory image to within minutes of onset, even as far as pre-ambulance, providing stroke type, size, position, and severity sufficiently well to support treatment much earlier than is currently even a dream. EMMA is a compact and mobile 3D functional brain imager. EMMA is safe, using orders of magnitude less power than the safety limits of a cell phone and no ionizing radiation. She is non-invasive and requires no contrast agents. EMMA is fast, enabling initial stroke diagnosis and later monitoring between confirmatory images. EMMA is orders of magnitude less expensive than current imaging gold standards. In an exploratory clinical trial, EMMA was 100% sensitive and 97% specific against the all-important hemorrhagic exclusion claim. EMMA detects hemorrhage as small as 0.13 milliliters, or roughly 5 millimeters across. CT for hemorrhagic exclusion is estimated at best around 80% sensitive for such a small hemorrhage. EMMA can directly color unhealthy tissue. EMMA's base technology can be extended to other pathologies, such as traumatic brain injury and other areas of the body. Time-sensitive neurological injury is merely EMMA's first target. EMMA's intelligence is a colossal multi-computer hosted in the cloud, remoted via the cell phone network. Only EMMA's head is physical, a cylinder with 128 transceivers that measures bioelectromagnetic disturbance, which converts to a 3D relative permittivity. EMMA's technological breakthrough reduces the astronomical processing time of a full-wave bioelectromagnetic tomographer from millennia to a few minutes. Simply unfeasible before today, and without numerous EMTensor proprietary optimizations to the physics, materials, mathematics, and software to processor mapping, EMMA's ability to color hemorrhage red and ischemic tissue blue in a DICOM image is a good bet. On the left is a CT of a hemorrhagic example taken from our exploratory clinical trial. I'll put up a little laser pointer so I can move around and show that a little bit better. So what we're talking about is in this region here. Let's see, yeah, this region here and here. This bright spot is hemorrhage. On the right, we have the EMTensor brain image, and the red spot there is where we've colored our suspected hemorrhage. In the middle bottom is a histogram of that red area compared to the gray bars would be the exact same area on the other side of the brain. What we notice is that there's a dramatic move to higher relative permittivity. Now, to give you some idea, blood is a higher relative permittivity compared to white and gray matter. That's why it would move that way. Then in the bottom left, we do basically a scatter plot of that same relative permittivity in the real imaginary parts taken from our image, and we put them on this scatter plot. And what we notice is basically a peak right around blood. That is a classic characteristic of a hemorrhage in our image modality. So we're able to point that out within, it takes about 0.4 of a second to capture the data and two minutes to make it available. And we can directly point the physician's attention to that red region rather than having them slide through many, many slices in a DICOM image. So, next slide. On the left, in this case, is an MRI image for an ischemic patient taken within a couple of hours of onset. So this bright green area is an ischemia in the diffusion image. Similarly, there's a little bit of a bright spot in this diffusion image. That's an infarct, a little bit of dead tissue. And this patient was thrombolized at 2.5 hours. So on the right here, we see an image of 9 hours where we still see the evidence of the ischemia. It hasn't quite cleared up yet. And we color it in blue. Then we do a histogram down the bottom here, and we can compare the blue areas on this image in blue bars to the gray areas, which is the exact same region on the other side of the brain. And again, what we see with an ischemia is that it dramatically moves the relative permittivity to lower relative permittivity. It's basically the blood has been removed. So it turns white or gray matter into something that is kind of below the relative permittivity of white or gray matter down here in the bottom corner in this scatter plot. So all ischemias have this characteristic. We did 43 ischemias and 9 hemorrhages, and every single one behaved in this way. So MS basically snapshots and provides an image within a few minutes, whereas an MRI, this particular MRI, would take more than 45 minutes just to acquire the data, let alone set the patient up for the appropriate IV to be able to inject the contrast agent. All the while, during that 45 minutes, the patient's brain is dying. This is the same patient. Now you notice at 28 hours, his ischemia is cleared up. He's still got a slightly worse infarct. On our image, the ischemia is cleared up on the right here. Still a few blue bars, but it's not an excursion right down to the bottom lower relative permittivity. So it's like some of the problem is still there and some isn't. And you see it on the scatter plot. You've taken that region that was all the way to the left of the white and gray matter, and some of it has moved back into the white matter region. So even some of the blue in this particular image is actually recovered tissue. Okay, so founded in 2012, EMTensor has 23 issued patents and more than 22 pending. It's based on 20 years of research described in more than 40 co-founder publications. Blood concentration has been connected to relative permittivity. We proved that through, and it's almost instantaneous. If you close off a pig's artery, you'll see it immediately. Research was funded by grants from Carolina's healthcare system, US NIH, and Austrian FFG. Our human clinical trial involved 52 patients and published in 2021 at ESOC in Helsinki. Our intention is to accelerate productization of EMA and multi-center clinical trials in 2022 and every subsequent year. You see lots of patients in there in trials, lots of new units, and lots of FDA approvals. Our team has 3 key elements: stellar guidance from a well-renowned advisory team, including physicians, medtech, regulatory, and healthcare leaders, a powerhouse problem-solving team of 7 Austrian PhDs, and leadership with astonishing growth track records, including engineering, program management from major strategics, marketing and sales and business development from healthcare. Together, this team has driven EMA's efficiency, mobility, and accuracy to astounding physician excitement over the last year or so.
Great, Stephen. Thank you so much for presenting. It's great to learn more about EMTensor. This is really interesting technology to me, of which I think I look at it and so much of the, what do I want to say? The physics of it all is, something that probably I never learned, I'm guessing. But tell us why others haven't done this before.
They've tried. Most of the major strategics have tried, and they tried about a decade ago. And they failed for a variety of reasons. But it's basically because if you try with currently available technology, you would conclude that it would take a millennia to process the data to get to the images that we produce. So what we've managed to do is change the physics, the materials, the way the mapping does onto processors, and we've leveraged this enormous, to give you some idea, the computer that we use, the best graphics processing unit on planet Earth is an NVIDIA A100. It is 5,000 processors, whereas you might have, I think you got 30 in your phone, right? And we use 200 of them. That wasn't even dreamt of even a decade ago. That's an absolutely ludicrous amount of processing. But it requires that to solve the bioelectromagnetic problem in the kind of detail that we need. So everybody said it's impossible. And we said, might be possible in the future.
We can do it. That's what you said. And then you did it.
Yeah.
All right.
That was even a year ago. So, we're now at 2 minutes.
That's awesome. We have a lot of great clinical questions from our audience, so I'm going to move on to those. The first is a couple about where is this going to be used? Is the idea that this is used at the bedside in the ER? Is this used in the field? Is it used in the patient's home by EMS? Talk a little bit more about that.
That's a great question. So there are 4 key areas. And we'll start with the stroke ICU, and we'll start with the notion that you can use it to just monitor the patient and protect them from the complications that occur and actually kill a lot of patients in the hospital. Right? And the reason for doing that there is that stroke physicians will give us a thumbs up and they'll infect the rest of the world. The very next place we use it is in the ER, and we help the ER decide which patients need to go for a confirmatory image, CT or MRI, which patients need to go home, and we weed out the ones that are real strokes from the ones that are mimics. Big benefit, huge problem, especially in the winter, I was hearing in Stanford.
Sure.
Then the next one is where it really rubber meets the road and it changes the world, right? We can deploy this thing because it weighs 50 pounds. You can pick it up, you can take it with you from an ambulance, just like you do with an EKG right now, and you can use it at onset with the patient before you ever put them in the ambulance. We talked to EMTs about this. They love it. It's exactly the equivalent of the EKG they take for sudden cardiac arrest or cardiac issues in general. They can very quickly figure out which kind of stroke it is, take the patient to the right hospital. If they need a thrombectomy, go there. If they have a hemorrhage, go to the nearest one. If they have an small vessel occlusion, go to one that can handle that. They can also potentially, if we prove this, we're already better than CT at being able to be sensitive to hemorrhagic exclusion. That means that it could become, in the not too distant future, a reason for justifying the application of TPA, the clot-busting drug. And that would make a dramatic difference to so many people's lives. It's already been proven that if you deliver a CT-based ambulance to the onset, you save 25% more lives, and you halve the figures for all of the disability metrics. In fact, it's been suggested that you get 2.5 times more zero outcome strokes than if you don't do.
That's
and
incredible.
The last place is in the diagnostic center, where it might serve a whole slew of different blood-related issues over time. But we have a little bit less information on that one.
All right. That's fantastic. A couple more clinical questions, because I love those. So how good is it at identifying old blood versus new blood? So do we have any information on sensitivity and specificity related to that? And then the accuracy and or sensitivity for detecting microbleeds is another question.
Yeah, so the microbleeds was actually that specific 100% and 97%, because we saw even in our clinical trial several 5 millimeter hemorrhages, which are basically where you sort of kind of define as a microbleed. Things above that are bigger than that, but microbleeds are down to 5 millimeters. And many of the physicians they work with actually want to separate microbleeds from non-microbleeds, because they want to not give patients TPA in the non-microbleed case, but they do still want to give them TPA in the microbleed case, because it's actually not a good enough reason to withhold it. That's been proven. There was a 570-person study done in Europe on that, and it's maybe a 2% difference. So it's really not a good thing to, stop a patient from getting TPA on the basis of that.
And the other question. Go ahead.
So why don't you repeat that? Because I think it was a 2-part question.
It was. So the other question was the ability to detect old blood versus new blood.
That's a very good question as well. We think that we can see in the same way as an MRI, it changes over time as blood ages. We can see that in the relative permittivity. Now, it doesn't show up in the real part of the relative permittivity. It shows up in the imaginary part, which is actually arguably more sensitive, but it's also more non-anatomical. People look at those and they go, that doesn't look like anything like a brain, but it's real for the physics of the situation. So we have to do a lot more patient studies to actually make an association. But what we're doing right now is we're basically registering MRI and CT and then comparing our results to those, so that anything that can be seen in any of those other modalities, we can set as a goal for our recognition algorithms to say, okay, can we see that with any kind of sensitivity and specificity? So it's over time, as we add more cases, we'll be able to answer that kind of question. It's just
Sure.
with 52 right now, it's just enough to say, hey, this is very promising. It's not enough to say, I can prove that's infarct because it's this, or I can prove it's old blood because of that.
Yeah.
Hugely important question, because if you tell the
Yes.
age, you can make a big difference to, okay, do I treat or do I not treat? What
Of
do I
course.
do?
Yeah. Well, Stephen, there are several more questions in the chat, and I hope all of you will stick around for our post-event networking, because Stephen will be there to answer the rest of your questions. Thank you so much for being here tonight and telling us all about EMTensor. This seems like really exciting technology that could be used in many, many different circumstances and indications, I'm guessing. So thank you for being here.